Okay, so let's go start with the afternoon session. So we have the pleasure actually to have three outstanding speakers for this session. The session is called the Development Brain Research. So we are going to still explore studies where they talk about evolutionary concepts, but it's going to be a lot also about developmental biology, focus in the brain, in the cerebral cortex. So the first speaker, well, it's a pleasure to have him here because he's a world expert in cerebral cortex development, is Victor Borrell, who is a researcher, a professor at CSIC and Universitat Miguel Hernández. And the title of his talk is the cellular and genetic mechanisms of cerebral cortex development unfolding. So thank you very much, Victor, and please join me and just give him an applause. Thank you.
Thank you. Thank you, Alberto. I want to first, of course, start by thanking you, Jordi, and the Institute of Neuroscience in Barcelona for the invitation. It's a great pleasure to be here, to meet some colleagues and friends and many new people. Yes, so basically what I want to talk to you about today is brain folding. And in fact, one of the most salient features of the human brain is that it has folds on its surface. And this is widely used in logos in many occasions when one wants to portray the human brain. And in fact, yes, it's not only the human brain of all of us that is folded, but also that of the majority of mammals, in fact. Folding of the cerebral cortex is something that occurs during development, and particular folds of the cortex are linked to particular cognitive functions or just functions in our brain. And in fact, when this development happens not correctly, then we're going to have very severe neurodevelopmental disease that usually involves effects as severe as learning disability and even drug-resistant epilepsy. So my lab has been interested for some time in understanding what are these mechanisms of cortex development that are particularly linked and important for the folding of the cerebral cortex. So the development of the cerebral cortex begins with a primary type of progenitor cells, are called radioglia cells that form this inner germinal zone called the ventricular zone. These progenitor cells, they divide, producing more of the same type of cell, and eventually they start making new types of cells. And these new types of cells begin by making intermediate progenitor cells, these other cell type that I'm drawing here, and these intermediate progenitors will now form a second germinal layer called the subventricular zone. And then eventually it will be these two populations of progenitors that will be responsible for producing all the excitatory neurons of the mature cortex. This is what happens in species with a small and even smooth cortex like the mouse that most of us work with. And then in species with a bigger brain, with a bigger cortex like humans, the basic schema is very similar. We still have a ventricular zone, a subventricular zone. But in this case, the subventricular zone is much thicker than in mouse. And in fact, is not only thicker, but also more complex histologically. Is distinguished in an inner subventricular zone and outer subventricular zone. Why is it thicker? Because it contains many more of these intermediate progenitors. It contains, in part, some of the same types of intermediate progenitors like mouse, but also it contains other types of progenitors that in mouse are largely missing, and some of which we have contributed to discover over the years. And we know that some of these cell types are important for folding of the cerebral cortex, including, for example, basal radioglia, here depicted in orange. So as I said, over the years, we've been making a number of contributions to our understanding of what are the basic elements that are important for cortex folding and how they function to induce the folding of this cortex. Our work has been based a lot on using the ferret as an animal model because it has a number of advantages that I'm not going to enter into here. But a lot of the work that I'll be presenting today is actually coming from using this animal as a model. And one of the observations we did a number of years ago is that in this ferret cortex, in the young animal, when folds just begin to appear, but it's clearly not as folded as in the adult, we saw a heterogeneous distribution of dividing cells. What you see there in white are the nuclei of cells that are undergoing cell cycle, and we saw that there were some regions with a higher density, like here or here, and other regions with much lower density. And this difference in density of proliferation coincided in space with the eventual location of this splenial gyrus that we see back here, and for example, the lateral sulcus in here. How we know that this is going to happen there? Because in the ferret, the pattern of folds is very simple, is very reliable, always happening exactly in the same place from animal to animal, so we can perfectly predict what's going to happen. And so we know that this region with this high proliferation is going to expand and develop as a gyrus. So with the intent of understanding what may be the genetic mechanisms that may regulate the formation of these folds, we decided to look at the transcriptomic differences between these regions leading to the formation of a gyrus and a sulcus. We decided to focus on this occipital part of the cortex. This is a sagittal section, this is olfactory bulb, because here is where we have one of the largest folds in the ferret cortex, this splenial gyrus, and for comparison, we looked then at the adjacent lateral sulcus. And we found several thousand genes that were differentially expressed significantly between the gyrus and the sulcus in these three different germinal zones. And in fact, for some of those genes, we found that their expression that was different between gyrus and sulcus was not only that, but it was quite modular. We had like boxes of cells with high levels of expression, and then a nearby region with cells with low levels of expression, and so on and so forth. And this led us to come up with a notion when combining data from a number of genes that in fact there is a whole protomap of gene expression patterns alternating high and low levels of expression that indeed anticipate and kind of predict or instruct the eventual location of folds and fissures in the mature cortex of this ferret. So once we had identified and shown that there is a transcriptomic protomap that predicts exactly where individual folds and fissures will develop in this ferret cortex, the next obvious question was, how is this protomap implemented? Who decides on how, where these genes have to be expressed? So one of the ideas that we came up with is that maybe there are epigenetic mechanisms regulating the establishment of this protomap as one of many possible mechanisms that regulate transcription. And among the many epigenetic marks and mechanisms that are known, we decided to focus on the acetylation of this residue in histone H3, because it's known that high levels of acetylation of this histone are linked to high levels of transcription. And we are not experts in acetylation, but so we decided to team up with the lab of Vijay Tiwari at Queen's University in Belfast, who really are experts at the epigenetic analysis of the developing brain. So what we did was to focus on stages before the protomap is actually visible. So we identified it in animals at birth, at P0, and we could still see that transcriptomic protomap at the late embryonic stages. So we decided, let's look a little before that to see what's happening before the actual protomap is visible, if already we have differences in this epigenome. And so we obtained samples at those different embryonic stages, and then we compared the transcriptomic profile, the epigenomic profile, between gyrus and sulcus. As you can see here in this PCI analysis, there was a clear distinction in the epigenomic signature between germinal zones in the gyrus and germinal zones in the sulcus. We found these differentially acetylated peaks different between gyrus and sulcus, present in promoter regions, exons, introns, and also intergenic regions. And we also found that for a number of genes that were related to these regulatory elements with differential acetylation, some of those genes were transcription factors, and we found that some of those transcription factors not only have their regulatory elements differently acetylated, but also they're differently expressed themselves at the level of transcripts. And among these transcription factors that had a positive correlation in these two parameters, we decided to focus on CAX2. Because CAX2 is a transcription factor that's been well known to be important in cortex development, although it is known to be important in later stages, in more the phase of differentiation of neurons and establishment of connections. So we could, I'm showing you here the raw data showing the level of transcript. There's differences between gyrus and sulcus in the two germinal zones for CAX2, and also there's different levels of acetylation of this histone H3, higher in the sulcus than in the gyrus. And we could here confirm the different levels of transcription for CAX2 in the tissue in these developing ferrets. Okay, COX-2 is a transcription factor, and as all transcription factors, it has a specific DNA sequence where it prefers to bind. And that's what's called a transcription factor motif. So the COX-2 motif is this sequence of letters here. So we search whether we could find this motif where COX-2 is going to bind in all the other peaks that are different acetylated between gyrus and sulcus. And as you can see here, we found thousands of these differently acylated sites that contain this COX-2 motif, which correspond to, in the order of 1,000 potential target genes, targeted by COX-2. Interestingly, also, as you can see here, these peaks were not only very abundant, but were differently abundant between gyrus and sulcus, particularly in this layer at E34. We found many more of the peaks that COX-2 can bind in the sulcus than in the gyrus. And that, again, corresponding with the number of potential COX-2 target genes. They were much more numerous in the sulcus than in the gyrus. Importantly, also, a lot of these genes that contain these differently acylated regulatory regions were also differentially expressed in the transcriptome, suggesting that maybe this different acetylation that COX-2 can bind may be responsible for their different expression. We looked at the ontology of these differentially expressed genes and differently acetylated, and you can see that they're linked in a number of terms to neurogenesis and early cortical development. So essentially, what I'm telling you is that we identify this transcription factor COX-2 that is differently expressed between gyrus and sulcus, and that it will bind to regions that are differently acetylated between gyrus and sulcus, pertaining to genes that we already know that are already differently expressed. So making COX-2 a very interesting candidate gene as kind of a conductor, an orchestrator of all this transcriptional program, potentially then regulating the transcriptomic protomap. So what we did was to manipulate the expression of COX-2 in these developing ferrets. We performed in utero electroporation of ferret embryos to overexpress COX-2 or the control plasmid, and then we waited 3 weeks until the cortex is developed, the folds are formed, to see if we find any difference when we have extra COX-2 or we don't. And I hope you can see here in these images how in the control animals where we simply overexpress GFP, there is no difference in the pattern of folding. And here, when we overexpress COX-2 here locally, you can see the emergence of a new fold and a new sulcus, where in the contralateral hemisphere, there is nothing. So just the expression of this single one gene in the progenitor cells is sufficient then to drive the whole genetic program that is involved into eventually forming this new fold with all its layers and its correct structure. Of course, we wanted to understand how is COX-2 doing this, because we put it in the progenitors, how we go from that to this. So we first decided to look into what's happening at the very early phases of development. So we repeated the experiment, but in this case, we waited only 3 days to see what's happening in the progenitor cells that are expressing COX-2. And to know what's happening to them, we performed single-cell transcriptomics of these GFP FAC-sorted cells. And what we found is that, of course, as in all single-cell transcriptomics analysis, we found a number of clusters of reflecting types of cells. And this is the clusters that we identify here. We have some progenitors, radioglial cells, and excitatory neurons. These are the distribution of clusters in control embryos, and this is in the COX-2 overexpressing embryos. And well, the first thing is you can see that pretty much all the clusters are in both cases, but the abundance is very different. On the one hand, we see that this pink cluster corresponding to those intermediate progenitor cells has been dramatically reduced in the COX-2 overexpressing embryos, where the population of neurons corresponding to about 30% of cells now is more than almost 50% of the cells. So we have increased the number of neurons, but with fewer of these intermediate progenitors. Now, this is a classical situation where we would conclude that these electroporated radioglia, instead of making neurons by producing those intermediate progenitors that I presented in the beginning, what this radioglia is doing is generating neurons directly, skipping the intermediate progenitors. And this is how we will have, with the COX-2 overexpression, much fewer of these IPCs, but instead many more neurons. So it seems that COX-2 may be inducing the formation of that fissure of that fold by inducing direct neurogenesis. Now, another observation we did here is that radioglia cells were not only in one cluster, like IPCs, but actually there were multiple clusters, which caught our curiosity to really look more into detail of this, because as far as we know, in mouse, there's only one type of radioglia cells. Why do we have 4 different transcriptomic classes here? So we decided to continue with the single-cell analysis of the developing ferret, but here we came with a little problem. Traditionally, and up to now almost, all the single-cell transcriptomic studies done, what they do is they just take a chunk of tissue, they run it up, they get a suspension of cells, they sequence, and then they identify the particular types of cells they find based on the expression of canonical markers. Oh, if it expresses TBR2, that's an intermediate progenitor. If it expresses PAC6, it's already a glia. But as I showed you in the beginning, in this species, we have a very large variety of progenitor cells, and some of them are quite similar to each other. So we needed to know exactly where is the location where we are analyzing those cells, because otherwise we may be missing information. So what we did in this case was to, as I said, for example, we have radioglia cells that are present in the ventricular zone as apical radioglia and also in the outer subventricular zone as basal radioglia. And these may be very similar, and so we may skip their difference if we just look at the canonical marker. So the solution to this was made by a really outstanding PhD student in my lab, Lucia del Valle, who did a phenomenal work in her thesis, where we started by, she started by microdissecting individually and isolating these germinal zones. In the case of this embryonic stage, only the ventricular zone, and then in the postnatal animal, analyze only the VZ, and then separately also the OSVZ. So we could know exactly whatever comes out of these are apical progenitors or they are basal progenitors. And also Lucia took care of microdissecting separately the gyrus from the sulcus, so we could also distinguish if there are differences in this developing cortex. So this is the summary UMAP of the more than 100,000 cells that Lucia collected from this work. Each dot, for those of you who may not be familiar, each single dot corresponds to one single cell, and what you see here is the transcriptional space of all the variety of cells that we have. We identified by looking at some, again, maybe canonical genes, we can identify excitatory cells, excitatory neurons, intermediate progenitors, and radioglia cells. When we looked at these clusters again, we identified some radioglia cells that were in the G1 phase of the cell cycle, others were in other parts of the cell cycle. Of course, these are cycling cells, and the transcriptome varies during the cell cycle, and this is something expected, but it caught our attention that actually we had 3 different clusters that seem to be in the same phase of the cell cycle, suggesting that maybe they could correspond to 3 different types of radioglia. In any case, when we quantified and looked at the relative abundance of all these clusters between the different conditions, between different ages or layers, or between gyrus and sulcus, we found the majority of these clusters are just equally present and represented everywhere, which was, kind of, not what we were expecting. We were hoping to find some difference, no? But essentially, almost everything was the same, except for these 2 clusters here, what we call cluster number 8 and cluster number 16. This one being enriched in the ventricular zone, and this one in the outer subventricular zone, suggesting that maybe these would correspond to the basal radioglia in the outer subventricular zone, and these to apical radioglia here in the ventricular zone. So we decided to pay a little more attention, particularly into these 2 clusters. So we analyzed only separately the cells coming from these 2 clusters, and this now is the UMAP of this subclustering. And we started looking at what are the characteristics of these cells. And when we looked at their original cluster of where they're coming from, the 8 or 16, that now we call alpha and beta, we see that they were still separating perfectly from each other, which supports then the view that these actually correspond to 2 classes of radioglia cells that initially I said cluster 8 and 16. Now I'm going to call them radioglia alpha and beta. So at least we have these 2 classes. Then we looked at those that we isolated from the sulcus and the gyrus, because we had dissected them separately. Now we know exactly where all the cells are coming from. And we saw that, again, these 2 clusters seem to be very separate, and you have here the quantification, which allowed us then to say that actually there is alpha 1 and alpha 2 radioglia, and the same for the beta, we have beta 1 and beta 2. The alphas being, the ones being enriched in the sulcus, and the 2s being enriched in the gyrus. Then we looked at the layer of residence. We have the VZ and the OSVZ separate. Let's look at that. Well, when we looked at their layer of origin, we found that they were actually completely intermixed. And you can see also here that there are essentially no difference in their enrichment, indicating then that these same 4 classes of radioglia cells exist not only in the VZ, but also in the OSVZ. Transcriptomically, we have a repetition of populations, although we know that at the cell biological level, they do have a difference. We looked at the characteristics of the transcriptomes of these cells, and we could conclude that the radioglia alphas, both the 1 and 2, are enriched in terms that are indicating that these are highly amplificative radioglia cells, where the betas, both the 1 and 2, are expressing genes related to more differentiation, which is, again, maybe linked to the direct neurogenesis concept that I was mentioning before. Now, if you paid attention in the quantification I showed before, you will have noticed that there is this one subcluster here, cluster S6, that was really very much enriched in the ventricular zone, and particularly also enriched in the sulcus of this ventricular zone. We were very curious about this particular subcluster. We looked at genes that might be expressed specifically in this one, and we found this one gene, CRIAB, that as you can see here in the heat map, is essentially only expressed by cells in that subcluster. Well, it turns out that CRIAB had been already described in the developing human cortex as being expressed strongly in the ventricular zone, and this was identified as being expressed by a new type of radioglia that initially was thought to be specific to the human cortex, called truncated radioglia. These kind of cells are exactly like the apical radioglia I showed you, but that process that they have extended to the surface is cut short and is so truncated. So what we were seeing here is that this truncated radioglia is not exclusive to humans, it's also present in ferrets with the folded cortex. And also we were seeing that these were highly enriched in the sulcus region because they were not present essentially in the gyrus region. For some of these cell types, for example, for the truncated radioglia, we looked histologically whether they really are real. If they're present, we could see them, for example, with antibodies against CRIAB. You see here the cell body and their basal process that is cut short. Usually this basal process will end up wrapping around blood vessels. And again, you can see here in a block of tissue, you can see the full span of the apical radioglia with their soma here extended all the way to the PL surface of the cortex. And here you can see an example of a truncated radioglia that is here with a short basal process that is also bent, as you can see there. Okay, so essentially what I've shown you so far in this part of our projects is that we have two main types of radioglia, radioglia alpha that are very amplificative and betas that are more neurogenic, that actually these are subdivided into 1 and 2 subclasses. There are some enriched more in the sulcus and some others are enriched in the gyrus, and they're clearly transcriptomically distinct. That these populations are repeated in the 2 germinal zones, the ventricular and the outer subventricular zone. And then, I have not mentioned this, but we found yet a 5th type of radioglia that is common in all these regions, layers, and parts of the cortex. It's not enriched particularly anywhere, and we call these radioglia gammas. And then finally, we have the super enriched in the sulcus truncated radioglia. One of the things that we were obviously very interested in is into knowing if this is something particular to the ferret, is just a curiosity, or is something that we may find also in humans, is something that was conserved during evolution. So we decided to compare across, you can see the phylogenetic tree of the primates and rodents and the carnivores. So we decided to look to compare our data set in ferret with public data sets from mouse and human. And so these are the 2 studies where we obtained the data sets that are done at stages of cortex development that are roughly equivalent to the stages that we had analyzed in ferret. This is the integrated UMAP of the cells from all these 3 studies, combining human, mouse, and ferret. And we can still see the main types of cells, progenitor cells, intermediates, and excitatory neurons. And here, when we focus in the radioglia cells that I'm showing you down here, we could clearly identify separately the clusters of cells corresponding to all the different types of radioglia that I showed before. Now, this led us to one important and difficult position, because we were describing many different types of radioglia cells in the ferret and saying that some similar types occur also in the human, but this contradicted what the literature said, which is that no, in the ventricular zone we have apical radioglia, and in the outer subventricular zone we have just basal radioglia, and they're transcriptomically distinct. Well, it turns out that these 2 classes, we were observing them in our human samples, but together both in the VZ and in the OSVZ. So interpretation was that, in fact, this is probably the result that one gets when looking at early stages of human cortex development, and this actually corresponds to later stages of cortex development, where these radioglia alphas are not existing in the ventricular zone anymore. They are not observed only in the OSVZ, and here they're replaced by these truncated radioglia. Okay, so to be convinced that a particular type of progenitor cell is really distinct from another, one wants to have some functional proof. And I think the best proof is looking at whether they have a different lineage, if they produce different types of cells. If they do so, they definitely will be different types of progenitors. So to look at the lineage of these cells, we could use our transcriptomic data to look at what's called the transcriptomic trajectory, which is an analysis based on simply the transcriptomic similarity between cells. So they're aligned along a course saying, okay, if a cell is almost exactly the same transcriptomically to the previous one, they probably are in the same lineage. They're part of the same biology trajectory of this cell. So we used monocle analysis to establish these black lines which determine these transcriptional trajectories, and this allowed us to identify 3 different trajectories or transcriptomic lineages, although that's not precise, from these radioglia cells. We found the trajectory number 1 starting with radioglia beta going into IPCs beta, then the second starting with radioglia alphas, producing radioglia betas and going on, and then this other trajectory. Now, very interestingly, then, this is from the whole data set. We wanted to know in the particular regions and ages what we had, which one of these trajectories was happening. So when we analyzed this from the complete data set, when we looked at the particular samples that we analyzed, to our surprise, we found that, for example, in the ventricular zone, both in the gyrus and the sulcus, we found the 3 trajectories happening. So this is at the same time, in the same place, we have these 3 different trajectories taking place. When we looked in the OSVZ, we found that that also was the case for that additional layer. It's definitely the case for the OSVZ in the gyrus, and in the sulcus, things seem to be a little simpler, not so proliferative, but in any case, we are seeing still the 3 trajectories happening again in one single layer at a single developmental time point, which is something totally different than what I presented you in the first slide, everything that we know from mouse studies, that things are super simple, radioglia, IPCs, neurons. But as I said, these are only transcriptomic similarities. We need to go further than this to really demonstrate that these are real cell lineages. Because a lineage is defined by a progenitor cell generating daughter cells, and they are really daughter cells from the mother cell. So to demonstrate that this is the case in our transcriptomic space, what we would need to do is to label individual progenitors. After a few days, capture all the daughter cells from that individual lineage that we can label and identify as unique, and identify that those cells really are daughters from the mother, and then analyze their single-cell transcriptome. Only this way we will know, yes, these cells with this transcriptome really are one coming from the other. Of course, this would be the dream experiment, and we were super lucky to really interact with just the right person to do this experiment, which is Christian Mayer at Max Planck Institute in Munich, who had developed a method called TrackerSeq. And this is simply based on the introduction in cells by a complex library of genetic barcodes that have a particular sequence. So when this library is so complex that when one introduces these plasmids, for example, by electroporation, the same combination of barcodes is almost impossible that happens in more than one progenitor. So we can recover the sequence of these barcodes, and we will see all the cells that are really coming from the same individual initial progenitor cell. So essentially, the experiment consists of simply electroporating this complex library and then allow this lineage to begin for a number of few days, and then recover the electroporated cells, and then we sequence the barcodes, and also we sequence the transcriptome of these individual cells. So this is just a summary of very complicated analysis. Here you see the different clusters that we found in the UMAP in the beginning, and these lines establish the coupling likelihood or probability, so the likelihood that cells in this cluster or this cluster will be forming part of a clone. If there's no line, we never found cells belonging to these clusters in a single clone. If the line is very thick, very frequently, we found cells belonging to those clusters being part of the same clone. And so with this, what we found is with this analysis evidence that the 3 different transcriptional trajectories that we had identified in the beginning really we are observing them as real cell lineages that are happening in vivo. So this was definitely proof of the existence of these 3 as real cell lineages in the developing cortex of these ferrets. Now, intriguingly, we observed another thing that happens when you get a lot of data, which is we saw a coupling also between these clusters of mitotic radioglia directly with these post-mitotic neurons, without intermediate progenitors in between. We looked very hard into it, and we're convinced that this is real, and so this would suggest that we are observing direct neurogenesis from radioglia to neurons, something that our transcriptomic trajectories had not revealed. And so we looked into this because, of course, I've told you in the first part that CAX2 is inducing direct neurogenesis in these ferrets. So we looked into which clusters may be expressing CAX2, and we found that indeed radioglia are expressing it, but not any radioglia, only the radioglia alphas are expressing CAX2. And the mitotic radioglia related to these radioglia alphas that are right here. So in fact, this CAX2 is being expressed exactly in this trajectory that we had noted before, but now that the tracker-seq experiments are revealing. And even more so, we have been able to see that this CAX2 is expressed in the basal radioglia alpha, not just the any radioglia alpha, not the apicals. So in the OSVC, and also particularly in the sulcus, as I started to tell you in the beginning. We looked then into these trajectories. Now that we're convinced that they're real lineages, we looked into the integrated data set with human and mouse cells to see if we could also identify these trajectories. The UMAP looks different, but it's exactly the same composition of cells. And here we could, with a trajectory analysis, we could recover the three lineages in the ferret. We could also see them happening when we looked at the human cells from this integrated data set. So in the human, we also observed these three different lineages. And then when we looked at the ferret cells within this the mouse cells within this data set, we could also only identify one of these trajectories. So in essence, what I've shown you is that we have in this developing cortex at least three different lineages happening in parallel in the ventricular zone and in the outer subventricular zone, that they are occurring simultaneously in the same developmental time, in the same region of the cortex. So they are somehow maybe redundant, maybe just complementary, and that we observe these three lineages happening in the species that have a larger and folded cortex, but not in the simpler and smooth cortex of the mouse. And just to recap again, I've shown you also that we've been able to identify a variety of types of radioglia cells. Some are more amplificative, and we find them enriched in the future gyrus region that needs to really amplify and make lots of cells. Some are less amplificative, more self-consuming, producing neurons, largely by direct neurogenesis, and this is driven, at least in part, by the high expression levels of CAX2, which then unfold the whole transcriptional and genetic program to make this happen. And with this, I'm done. I just want to mention again, the main people that did the work on of these studies mostly was, again, Lucia del Valle in the lab, but they helped with many of our people in the lab. Of course, I want to acknowledge our collaborators that made also possible to finish these studies, our sources of funding, and I'm happy to take any questions. Thank you for your attention.
Is this working? Yeah. Thank you very much for the talk. I have a very naive question. When you induce the extra folding, do you see any cognitive or behavioral changes in the animal?
We're looking into that. I don't have any data to talk about yet, but yes, of course, we want to look into it. But it's not as easy as it sounds. Well, nothing sounds easy, but we're doing tests specific for the region where we're changing this fold, no? Because these changes are not all over the place. In my lab, we have a tradition in looking at the visual cortex and the visual behavior and experience. This is more in the prefrontal or motor area, so we have to adapt to that. But yeah, hopefully we will get some data soon.
Hey. So you say you have different types of radial glia, then and now you are also doing lineage tracking in Vivo. So I'm wondering, did you have a look at the neurons that are generated from these different types of radial glia? And then do you know if they are in some way similar, or do they have the same morphology, or are they very different? Mm-hmm.
Yes, thank you for the question. So we have looked at this, but only very early. So basically from the same data set that we have, we already see immature neurons, even if we're looking at the germinal zones. But for example, the OSVZ, there's already migrating neurons there. We also capture those. But the signatures that they have are of very immature neurons. And from this data set, all neurons seem very similar. We do see differences in the maturation trajectory between gyrus and sulcus. We see these, but so far not between the different lineages and the different types of radioglia. I suspect that they will eventually be different neurons, so we're working now into analyzing at higher layers of the developing cortex to see them when they're more mature, because I suspect that since we're only looking at the transcriptomes, there may be signatures in their epigenome or in their chromatin accessibility that will only show us transcriptomic differences later on in the maturity of these neurons. But so far we have not seen any differences. Yeah.
Thanks, Victor, for a great talk. Do we have, perhaps we don't know enough, but do you make predictions about human pathologies associated with the subtypes of, or the different lineages that you have identified?
Yes, we looked at this. So we've looked at genes, all the genes known to be related to developmental malformations of the cortex in humans, and there's many different developmental malformations. And we have not been able to identify differences in expression levels in types of radioglia, but yes, we've been able to identify differences in the regions. For example, higher expression of microcephaly genes in gyrus, higher expression of lissencephaly genes in sulcus. Yeah, so that's in the paper, is in supplementary data, you can look at it. But yes, we do find differences in expression levels of these, but unfortunately not at the level of types of radioglia. Again, a similar answer as before. Maybe it's unwavering, but maybe the detail of our analysis or the depth of our sequencing is not sufficient to find differences that may exist there if we go deeper.
Thank you.
That's a very good question. There were a couple of questions in the front.
Couple of questions here.
So thank you, nice talk. So I have one question because it is related to this output, no? With the news that you were saying before. So I was wondering about this alpha and beta glia. Do you find them also at very early stages of development, just at the beginning, or they appear through development?
So this is something that we also have ongoing now. So what I showed you is what I know today, which is E34 and P1. So P1 is when upper layer neurons are being made. E34 is maybe when layer 4 neurons are being made. It's not super early. If you're thinking when radioglia mostly amplify, we need to go much younger, and we're working on that, actually. Yeah.
Here, Gabriel. Yeah, here.
Hi, Victor. I'm just curious. When you induce CAX2, you end up having more upper layer neurons, or did you check if you have more upper layer neurons and callosal projecting neurons in the prospective gyrus?
So we looked into this. We did not count cells because that can, you can get off very easily because of you do that based on histology and depending on how you section, which section you count anywhere. We looked at the surface area of the cortex and surface area of the different layers or contours. These are things that are easy to measure, and we did. And we found no difference. So our conclusion was that we are changing folding without changing the size of the cortex. So we're just changing the way this cortex is being made. How it is patterned. We're not making a bigger cortex or a smaller cortex. We're just changing the way the neurons are being made, if directly or indirectly, and that will have an impact maybe on how they migrate and how eventually that ends up folded. But only the shape changes, not the size.
And the connections?
And the connectivity, this we have not looked. No.
Okay, last question. Yeah.
Very short question. In terms of vulnerability, their origin is important. The vulnerability of these neurons is important if they come from the sulcus or the...
What kind of vulnerability are you thinking
for example, meaning if you check these neurons in an adult animal, for example, or I don't know, I was sensing that depending on the development, the migration, the differentiation, when they become adult neurons, they can be more or less vulnerable in terms of neurodegeneration.
Yeah. So maybe probably you know more than me about this. Yeah, unfortunately, I have nothing I can say here.
No.
Okay. Sorry.
Okay, join me and say thank you to Victor. So let's move to the second speaker. It's a house speaker. It's Dani del Toro. He's a researcher, now an associate professor here at the School of Medicine in the Hospital Clinic. And he's going to talk about the expansion of the neocortex and behavior performance by in vivo reprogramming. Thank you, Dani.
Thank you. Yeah, so it's also a pleasure to be here today, and thank you so much to the organizers for the chance to share with you what we have been doing using Yamanaka factors as a tool to increase neurogenesis in the brain. And I want to start with this scheme because, as you all know, these 4 factors, the Yamanaka factors, are very well known for their ability to reprogram cells to a pluripotent state. And that's why how we can obtain induced pluripotent stem cells, iPS cells. And nowadays we have several protocols to differentiate these cells to almost any cell of our organism. So this has been a revolution in science, but these factors, they can do more things. One study from Ipsua Belmonte's lab showed that transient induction of these factors, just few days in mice per week, is enough to slow down aging. And this they saw in just peripheral tissues, in the skin and in muscles. These 2 mice, they are litter mates, they have the same age. But as you can see, one looks much younger than the other one, and this is the one getting this transient induction of these factors. And since this study, several labs have shown that indeed, peripheral cells with a transient induction of these factors is enough to bring their transcriptomic profile back to a younger state. And this has been observed not just in skin or muscle, but also in heart, liver, and kidney. So here what we ask is what happens if we take these factors and use them in the brain? Is there anything useful? And at the beginning, we focused on development. And it's not as artificial as it seems to use these factors here, because there is already a pluripotent program activated already at early stages of this process. We can find neural progenitors expressing different combinations of these factors. We can see OCT4 together with SOX2, even NANOC. That is a pluripotent marker. So what we did was to take advantage of this mouse model developed by Manuel Serrano and Maria Bat, the 4 Yamanaka factors controlled in a Teton fashion. And that's great. It's a great tool because we can combine this animal with different mouse lines where we can drive RTTA expression with different promoters. And because we add doxycycline in the drinking water of pregnant females, we can control when, for how long, and how much these factors are being expressed. And this we did in 2 different models. The I4F ROSA, the 4 factors induced animal, and ROSA is because we target the entire embryo. We drive RTTA under the ROSA promoter, or nesting. So we target only the central nervous system. And we did different treatments during development to see what happens, what kind of phenotype do we get. And I want to start with this movie because at the beginning we wanted to know what happens if we push the system. So what do we get if we do a strong induction of these factors for 4 days between E10 and E14? So on the right, we have an hemisphere of a mutant embryo where we have this strong induction for 4 days. And on the left, we have the control littermate. And if we go through the stack, you will see that at glance, everything looks fine so far. So we see the cortex, we have a bigger cortex and striatum and also thalamus. So everything looks fine so far, but if we pay more attention, you will see how the germinal zone starts to invaginate. You see here? And this should not happen. This should be smooth as we can see in this control hemisphere. And why this happens? So this happens because if we stain the number of progenitors here in yellow, these are expressing PAC6 and also SOX2, we can see that after these 4 days strong induction, their numbers are massive. And it is so massive that the ventricle per se is enlarged by 50%. And there are so many that the only way to accommodate them is to do these invaginations to increase the surface of this layer. But there are most things, but here there are other things happening, not just that the numbers are increased. Look, in the control situation, always these apical progenitors express PAC6 as their identity marker. And as you can see, it's a very homogeneous staining. All cells look very green and red because they also express SOX2, so the merge is yellow, and it's very homogeneous. Now, with this strong induction, we have more progenitors, but they also, some of them start to lose their identity marker. We see less PAC6, and that's why in the merge we see these reddish areas. And this we can quantify. We can go cell by cell, so each dot is one cell, and the orange ones are the mutants. And as you can see, they are shifted to the left because the levels of PAC6 are lower. But this not only happens to progenitors, this also happens to neurons. So now let's go back to the same image. Here in magenta, we label the cortical neurons, population of them with a CTIP2. And as you can see, this layer is very smooth and homogeneous. With an inset, we can see here the neurons, and this layer is also almost devoid of SOX2 cells. Now, here you just can see that this magenta layer, it looks kind of disrupted. It's not homogeneous, and if we zoom in, we can see that neurons start in some regions to lose their identity. They are not expressing CTIP2. We only see SOX2, and even we can see sometimes PAC6 in this layer. And this we know that neurons are the ones losing their identity because we can see co-expression, for example, of NANOC, a pluripotent marker, with a CTIP2 that is a marker of neurons of the cortex. And we know that if we keep pushing for another 3 days with a strong induction of these factors, the entire brain becomes a mass of IPS-like cells where we see NANOC everywhere in all layers. And at this point, we ask ourselves, well, can we find a protocol or an induction of these factors where we get some benefits while preserving the identity and the tissue histology of the brain? And the answer is yes. So if we tailor the induction, we have a lower induction for very few days, we can get sections of the brain that looks normal. And now we can compare the low induction of these factors with a control littermate. Of course, the brain is bigger, but now you just can see how these apical progenitors, they nicely express PAC6. We don't see them losing their identity. And now we can start to see also in the areas where neurons are generated and they are migrating in the intermediate zone, and also in the cortical plate, we see this strong abundance of SOX2 cells. And this we can quantify. We have more SOX2 cells in the intermediate zone and also in the cortical plate compared to their control littermates. And this we know that is because neurons per se are expressing these factors. So they are expressing SOX2. And this we can observe, for example, in the cortical plate, where, as I said before, there are very few SOX2 cells in a controlled situation. And as you can see, these cells are not expressing neuronal markers such as CTIP2. It's like a binary option. It's either a neuron or is a cell expressing SOX2. But now, when we induce these factors, we can see that SOX2 cells are expressing neuronal markers. So neurons are expressing them, and this is tolerated. And this we can also observe by single-cell RNA-seq. That is what I'm going to show you now. So here we took the cortices of embryos with a low induction of this factor under control littermates. This is how the distribution of the cells in the cortex looks like in this UMAP. We have progenitors, apical in green or intermediate that also before Victor explained very well where are located. And then we have this big cluster of neurons and other cell types. But let's focus with these 3 big ones. This is how the control UMAP looks like. Now, when we do the induction of these factors, you can see that overall, the same clusters are preserved, and they look exactly, or they look extremely similar. But there is the appearance of another 2 new clusters when we do this low induction of these factors. And these 2 clusters represent neurons, that we call them as I4F neurons, and they also represent apical progenitors. And these 2 clusters, the difference is that their transcriptomic profile is more immature compared to their respective control populations. And also, they are enriched. They are expressing high levels of these factors, of the Yamanaka factors. For example, OCT4. And as you can see here, we can go up to 30% of apical progenitors expressing OCT4. And as you can see, it's very little in case of a control situation, and 15% of neurons. But this is transient. This perturbation is transient and is tolerated. And now what happens if we apply this treatment to the entire embryo? We get embryos which are bigger compared to their control littermates with bigger brains. But also we can do this only in the nervous system, only in the brain. And as you can see, if you look, the frontal part of this embryo is enlarged. The brain is almost doubled in size. And you can see that this head, it looks kind of humanized, let's say mouse head, because of this big expansion of the prefrontal cortex. And these animals are viable. So they get born, they reach to the adult, and now what happens with them? Well, at adult, what we see is that, of course, this expansion and this brain is bigger. we have on average around 25-50% more neurons, and this is mainly in upper layers, in layer 2 to 4, as you can see here. And it's not just neurons. Also, glial cells increased and go in the same proportion. So here, we just can see, for example, for astrocytes, their numbers increase, and it's mainly in upper layers, where we see this strong increase in numbers of neurons. But also because we have more neurons, now we also can see more myelin. So we stain for the myelin binding protein that also stains oligodendrocytes, we see more staining. So now what happens when we do behavior? So are these animals fine having this bigger brain with an expanded cortex, or is there any harm or any negative effect? So we did a huge battery of tests, and most of them, and I'm going to show you a couple, were negative. So we did not find major changes, which is good. So at least we don't do anything negative, and these animals are behaving as good as the controls. Two examples of these experiments where they didn't show major changes is the open field, when we check for the motility, both the ROSA, which is the entire embryo, which is bigger, or the nesting, just the nervous system, they did as good as their control litter mates. In case of anxiety, if we use the elevated plasmates, so here animals are anxious, more anxious if they are exploring the open arm, you just can see that the ROSA, they did as good as their control litter mates, and even the nesting ones, only where the brain is bigger, they even were less anxious. They were spending more time in the open arm. The test where we saw a strong difference that was for the better were the following. The motor behavior, so here we put the mice on the accelerated rotarod, and as you can see in orange, the mutant mice already from the beginning, they are better. Their latency to fold is increased. And this, when they reach their plateau, both lines, the ROSA, which is the entire embryo, or the nesting with the bigger brains, they do better compared to the control litter mates. And the last test was the social test. So whenever these mice, they have to choose between spending time with another mouse in a cage or an empty one, they prefer to spend more time with another mice. So in this sense, they are more social friendly. So these are the technical messages that I want to give you here, and is that we can use these Yamanaka factors to increase the proliferation. We can get more neurons, also more glial cells. We know that we are perturbing cell identity, but if we do this for a short period of time and with a tight control of their expression that is tolerated, cells will go back to the regular identity. We get bigger brains, and this we have observed that for mice, they also come back with an improvement in some tests, for example, for motor and social behavior. And I want to finish just by thanking a lot of people, of course, the members of my lab, so 4 PhD students today. There are 2 posters by Sofia, number 45, and Irene, number 46, where you can find more information about the Yamanaka factors. As you can see, we have 4 as the 4 Yamanaka factors. Then I would like to thank Rudiger at the Mass Plan because they did experiments with the I4F nesting line. Of course, I'd like to thank a lot Albert Giral lab, because thanks to him, we could do all these adult studies and setting up all the behavior, plus the option, because today we didn't have time to show, but also to apply these factors in the mature brain, in mature neurons to rejuvenate them. And finally, I would like to thank both Manuel Serrano and, of course, Maria Bad for getting access to their mouse model and for discussions about their programming. So, and of course, all of you for your attention. Thank you.
Thank you, Dani. Any questions for Dani?
Thanks, Dani, for a nice talk. It's incredible work. I was curious about, you mentioned that the area that was more enlarged was the upper layers of the cortex. This is because the time when you're giving the doses is that. And related with that, what will happen if you do the increase of the Yamanaka factors in another time? and what is happening in other areas apart from cortex?
Yeah, no, that's a really good question. And indeed, it depends a lot on the timing and also on the dosage. So the window is quite big. It's from E10 to E14. So one would expect also to have impact in deeper layers. But at the end, what we see, the major changes are in the upper layer. So we still don't know really what is. So we know that when that cells expressing these factors, they keep more time dividing, which also could be like another factor that could explain why, even though we start at E10, is mainly the upper cortical neurons, the ones showing bigger numbers. And regarding the other regions of the brain, yes. So the thalamus is also strongly affected. The hippocampus, if we do these treatments later as well. So depending on the time, different regions are more or less affected, which also fits with their developmental stage. Yeah.
Here, Victor. Yep.
Thank you. Congratulations. That was a fantastic talk. I was wondering if you have followed those animals in the long term to check about the onset of tumors, because that would be the main caveat.
Yeah, so the maximum time has been 5 months, 5 or 6 months, and they were fine so far. So all the histology was fine. So indeed, what is for me very interesting to observe is that even though during development, if you do stainings at, when we do the treatment, you can see strong effects. Just 1 or 2 days later, the brain looks totally normal. Of course, the size is different, but all the markers, they look pretty normal. And indeed, neurons are super efficient shutting down these factors. So in short, in adults, so far we haven't seen any tumor, also because we used the lower dosage that we know that during development is tolerated. So that's also another. If we increase, I'm pretty sure that we will get tumors. Yes.
Thank you.
There is another question here.
Dani, fantastic. I wanted to ask you, so the surface area of the ventricular zone is clearly enlarged, but the ventricular zone is thinner.
Yes.
Have you counted total number of cells? Do you know if you have really more?
Yes.
And why do you think the expansion is lateral, but then the reduction in the radial dimension?
Yeah, no. So yes, so because the ventricle is larger, if you count the cells in fixed width, you will count less. But if you count the entire perimeter, you count more. And why? And I think that it gets thinner because of this expansion. Maybe it's too fast, maybe at the beginning because we start at E10, and maybe somehow there is more growth in the lateral axis more than in the radial. So we don't know why it looks so thin, but maybe it's linked to this increased perimeter, which is very strong. Maybe these two are related.
So I have a question actually on this regard. So do you think that it's more like a tangential expansion? So you are reproducing the initial expansion of the neuroepithelia?
Yeah, that's also what I think, because if you see the thickness of the cortex is not much thicker. So it is a bit thicker, but it's not much. So the maximum expansion is in the tangential axis, is where we see the major effect, more than the radial. Yes.
There is another question over there, I think. And this will be the last question. Yep.
Okay, so it's 2 quick questions. You do an overall amplification of the Fourier-Manaka factors are expressed on the whole brain, and you see invaginations in the interior part, but not in the outside. Do you see any folding, or could you see that if you express it locally? And the second is these mice are also moving more.
If they are what?
If they are also moving more, not only being more social, but if they are moving more.
Yes. Okay, so regarding the first part with the folding, so in some animals, we have observed folding in the cortical plate, mainly with the nesting, more than in the ROSA, but this is not the norm. So it's not that all of them show this. So that's something that we are studying, whether instead of having an expansion in an homogeneous way, it's better to do this locally. Maybe this will help, and also link with the previous talk about this heterogeneity of cell proliferation. And then regarding the hippocampus, this invagination in the germinal zone,
we think that is because of the increased expansion in the number of progenitors, and just for fitting them, just for having more space and increase the surface, it gets invaginated. So this should not be like this, it should be smooth, and only the outside, no, at least for Cortex, it should be the one folded. Regarding motor, so with the nesting, it is true that that's something that now we are checking. It seems that maybe they could be better runners and run more also with the open field. That's something that we are also checking. So yeah, so it could be, so at the end, for the motor aspect, they are better in that sense, yes.
Okay, yeah, this will be the last question. So let's thank again Dani. So the third speaker of the session is Gabriel Santpere. It's a Miguel Servet investigator at IMIM, and he's going to talk about tracing the initial emergence of cortical disorder risk through gene regulatory network analysis. So, floor is yours, Gabriel.
Thank you, Alberto. And well, it is for me a pleasure to be here and share my research with this audience and with these speakers. Today, I'm going to talk about our program, our research to try to understand the implications of neuronal progenitors into the risk of several brain diseases. I will start by necessarily explaining into this audience that the brain is the result of a really long, complex, and protracted process that involves several events, several processes that are subsequently, that are, developing through a long time. Some of them occurs in utero, for example, neurogenesis, neuronal migrations, but then some extends way through adulthood and even entering into the third decade of life, for example, myelination of the axons or the synaptic pruning. So it is an organ that takes up to 3 decades to complete. So many of these processes that we see during development actually are at some point or other regulated by this set of molecules that we call transcription factors. And here we have a list of some transcription factors that have been related to many of these processes, either the onset or the maintenance of these processes, neurogenesis, migration, axonogenesis, or determining the identity or the differentiation trajectory or the final terminal differentiation or lineage commitment of cells. And then this is far from being exhaustive, just some examples. But in my lab, we focus on these many critical transcription factors for neurodevelopment with the idea of determining the gene regulatory networks that they orchestrate, identify the mechanism by which these transcription factors bind the DNA, the mechanisms of specificity of this binding, how they partner, and also how they remodel the chromatin to enable these transitions from cell to cell during development. Also, we have a special attention to evolution of these gene regulatory networks, see by looking at gains and losses of binding sites, how these changes in the gene regulatory networks enable lineage specializations or derived features in the evolution of the human brain or the mammalian brain, say the appearance of the corpus callosum, the cortex, or the unique human brain features. So, but today I'm not going to talk about this. I'm going to talk about our study of gene regulatory networks in the context of brain disease, how we can map the risk of brain disorders, the genetic risk of brain disease into these gene regulatory networks. So human brain diseases can have very different features in terms of onset, in terms of etiology, and also in terms of the subjacent genetic architecture. Some are more polygenic and contributed by low effect common variants, and some are more monogenic. And many have unique human manifestations. We make them very interesting to study under the point of view of evolution. So one approach to understand the etiology of these diseases from the genetic footprints could be to try to understand if these genetic causes identified by genome-wide association studies or Mendelian approaches are actually converging into specific cell types or into specific time points, or molecular pathways and gene regulatory networks. So this idea, I started to explore it more than 8 years ago when I had the chance to go as a postdoc in an ancestral lab at the Department of Neuroscience at Yale. And in there, I have also the opportunity to put my hands on this impressive data set of bulk transcriptomic data of the human brain across the entire lifespan from the 8 post-conception weeks in the embryo up to adulthood, 40 years old. So we have in total 600 samples from 41 donors of these 16 brain regions, including 11 neocortical areas and subcortical areas like striatum and thalamus and so on. So then we reanalyzed genome-wide association studies to try to see if the genetic risk mapped to a specific gene modules that were correlated to specific areas or time points. And by doing that, we found this module number 37, which was contained a lot of transcription factors in which they had been described, or we described by the reanalyzing of this data, associations of neuropsychiatric traits with regulatory elements around these genes. So for example, MEF2C is a transcription factor that contains a high convergence of different neuropsychiatric traits, and also TCF4 is another one. When we look at the enrichment of cell type, which cell type expressed more of this gene module, we found immediately that this was highly enriched in excitatory neuron signatures, which, surprise, right? They are neuropsychiatric traits, so here not much to learn, right? So this started the second project that I'm going to present this today, which is what we have been developing in the lab, which is try to understand the implications of progenitor cells instead of neurons into the etiology of these diseases. There is several work suggesting that the etiology of this disease can be traced back to alterations of the spatiotemporal identity of these progenitor cells. So even if the genes are expressed or even are top expressed in neurons, they can still be expressed in progenitor cells and exert their pathological effect in there. So this is very poorly understood. So we tried our goals in this project, in collaboration with Nicola Micali in Pascal Rakic's lab, was to map the disease genes into very early cell types in neurodevelopment, to identify gene regulatory networks in each one of these early cell types and see if they have been altered in disease. And then, for each disease, assigned like a critical phase in development for which their genetic culprits can exert maximum effect. So mapping the risk into development is essentially dealing with the complexity of this dance of transitioning cell populations that Victor and Dani has presented here before, in which we have these progenitor cells at the beginning that are amplifying, and then at some point the cells begins to divide asymmetrically and start first producing deep layer excitatory neurons, subsequently followed by upper layer excitatory neurons, and then finally these cells starts to produce glia, astrocytes and oligodendrocytes, and finally they lose their proliferative potential and become ependymal cells. So essentially they are the same cells changing the transcriptome as they mature, and as they transect or traverse this maturation process, they produce different output. So we would like to map the risk of this disease into this axis of progenitor maturation. All right, so to do that, we curated gene sets of a myriad of neuropsychiatric traits, the major ones like autism spectrum disorder, schizophrenia, bipolar disorder, depression. And also we complemented this set by genes associated to cortical malformations, which tends to be more monogenic, including microcephaly, lysencephaly, heterotopia, polymicrogyria. So I think with this set, we cover a really good deal of the different types of brain diseases or diseases that can affect human brain. So if we want to map the risk into these very early populations of cells in humans, there is a really big deal because, there are not so many data sets, good quality single-cell data sets of this very early stage. So we had fortunate that this was produced very recently under the context of Brain Initiative Cell Consensus, this Obama Brain Initiative invention in the States, in which essentially they produce 1.6 million cells, single-cell transcriptomes of the entire central nervous system of the first trimester in the human brain. So we get this data. And then the first thing we did is to try to see in this different disease that you can see here on the x-axis, what is the measure enrichment, just a very naive question, what is the measure enrichment of these gene sets into these different measure cell populations? And we can see immediately that for the neuropsychiatric states, many of these enrichments are driven by neurons, again, yes, not big surprises, except of Alzheimer's disease, for which we know that the genetic architecture of Alzheimer's disease points us toward microglia rather than neurons. This is already known, but is shown very clearly in this analysis. And also when we look at the cortical malformations, we start seeing enrichments in intermediate progenitor cells, for example, in microcephaly or polymicrogyria. And then we have also even the implication of radial glia cells, also in microcephaly and hydrocephalus. All right. So, despite of these measured enrichments, we ask ourselves, okay, these genes are measurably, for example, enriched in excitatory neurons. But do they also expressed in these neuronal progenitor populations? How many of them are actually there as well, right? So it turns out that up to 75% on average of all these gene sets are also expressed in neuronal progenitor populations. And you can see here that 100% are expressed in microcephaly. This is quite expected. But also looking at autism, autism is super enriched in excitatory neurons, but nevertheless, most of the genes are also present and are also expressed in these different populations of neuroprogenitor cells. The lowest one of them is Alzheimer's disease, but besides that, the rest of them are really, really represented among. So, going further to this thread, we question ourselves whether these genes could be also expressed even earlier in these subsets of progenitor cells that conforms these transient structures in the developing brain that are called the brain organizing centers. These structures that are transient, they are long gone when you are born, instruct the patterning of the brain during development. We have, for example, the cortical hem producing Wnt and BMP factors, instructing the dorsalization of the brain. We have the floor plate secreting sonic hedgehog and instructing the ventralization of the brain. Or we have the rostral patterning center producing FGF and extracting the rostralization of the brain, right? So in that data set from the human, there is no enough resolution in terms of single cell transcriptome of these brain patterning centers, but we could collaborate with Niccolò Micali in his project on the macaque, very early macaque samples, if they could retrieve cells with the identity of this brain organizing center. So we can see here in this UMAP some cells belonging to the cortical hem, anti-hem, or the rostral patterning center. So this is perfect data set to analyze the expression of our disease gene sets in here. Many genes were found expressed in these progenitor cells, but to our surprise, 25 of these genes were only expressed in these brain patterning center genes. So there are genes, for example, genes related to autism, which globally are enriched in excitatory neurons, yes, but in neurodevelopment, they are only expressed at these early time points in, for example, the cortical hem. So that is really intriguing and interesting to follow up in functional studies, because it could imply that the patterning, the very early patterning of the brain can have dire consequences and develop neuropsychiatric disorders. So it is important also to understand that this window that we are studying is very inaccessible, particularly in humans, so it is important to have good in vitro models, and there has been a lot of interest to develop brain organoids and 2D cultures to model these very early stages. So with Niccolò Micali, which is essentially experimentalist, he had characterize very well his system of radial glia maturation in vitro in 2D cultures, in which essentially by passaging neural stem cells, he can, and controlling the concentration of FGF2 factor, he can essentially recapitulate the different phases of radial glia maturation that we observe in vivo. So we have, for example, some very early NPC stage that resembles organizing states, followed by early or late neurogenic radioglia states, and ending up with the gliogenic states. So we have very nice in vitro system to analyze now if the expression of our genes from different diseases actually resembles that we found in vivo. And the answer is striking yes. So the correlation is very high in between the two systems. So we have essentially even more genes expressed in this in vitro system, because these are bulk, we have more characterization of the transcriptome, but nevertheless, the correlation is very, very high. That's good for the modeling part. So then we proceed by analyzing the expression dynamics of these individual genes from each one of these gene sets across the different diseases. For example, we have here microcephaly, in which each row is a gene related to microcephaly here. And then here, these genes are sorted by their expression in this in vitro radial glia maturation system. So we see many genes that peaks at the very early stage of radial glia, and genes that peaks at the gliogenic stage of radial glia. Second, we analyze the expression of the genes in a dataset that allows us to query if the gene is more expressed in progenitor cells compared to neurons. It's kind of a single cell differentiation analysis in which we have stem cells and we have neurons, so we can have this axis of neurogenesis. So if this is red, it means that the gene is more expressed in progenitors. If it is blue, it means that the gene is more expressed in neurons. And then finally, we query this other axis, which is whether the gene is more expressed in immature neurons or it is expressed in more mature neurons. So we have, once you are a neuron, if you get, a maturing identity to So with these three axes, so the radial glia maturation, neuron to progenitor to neuron, and neuron maturation, we could see immediately that there are differences between diseases. Microcephaly are more enriched in early phases of radial glia maturation and also in more progenitors than neurons, but focal cortical dysplasia, on the contrary, is more enriched in neuronal and in mature neuron profiles. So our objective here is to compare these different diseases and see if they have different windows, right, of vulnerability of these different axes. So here we aggregated over all these genes, like in a stack plot in which you see the distribution of how the gene peaks into the different phases. And we see that microglia have effectively, we have a number of diseases that are, that the associated genes peaks into early phases of radial glia, other diseases that they peak in the neurogenic phases of radial glia. For example, ADHD or bipolar disorder, some in a later phases of neurogenesis, like ASD, and finally some diseases that peaks in the gliogenic phase of the radial glia. If we do the same exercise on this other axis, on the progenitor neuron axis, or in the newborn neuron, mature neuron axis, we see also interesting findings. So, for example, ADHD is more associated to mature neurons or schizophrenia. However, for example, ASD is more enriched in profiles related to immature neurons. So the window of vulnerability is more transient. Okay, so now it's time to understand the regulatory logic that exists within each one of these disease gene sets. Try to identify the transcription factors, if any, that co-regulate these gene sets. So it's time to define what we call regulons, and we tried two different approaches. We tried many, but here I just present two different approaches that we tried. One is, very naive one, is in one disease gene set, exists any transcription factor within the list that is enriched in binding sites in targets that are also within that list compared to the random expectation? That's one. And then the second approach is more sophisticated, is try to take RNA-seq data and chromatin accessibility data in each one of the cell types, single cell, and then try to build cell type specific regulons, building networks that we can perturb in silico. Okay? So for the first one, for the disease regulons, this naive approach, we happen to find 29 of such instances in which we have transcription factors enriched in targets that belong to the same gene set list. And if we order these core transcription factors that we called, these core 29 transcription factors across this axis of radial glia maturation, we see some transcription factors that peaks very early and some that peaks very late. And by this strategy, we can kind of factorize diseases, gene sets belonging to diseases, to subnetworks of this disease based on different core transcription factors. That's one thing that we could do. So we found distinct pathogenic networks within one disease. But we also find that same genes, same core transcription factors, can have a huge crosstalk with other disease transcription factors. It's like a brain pathogenic network, let's say. And they are sometimes very well connected. For example, MEF2C is in the receiving end of many of these disease transcription factors. It's really convergent in terms of regulation, as you know, as we have anticipated from the bulk RNA-seq study. Also KLF4. Now, for the second strategy, let's make this global reasoning that I believe now we are a little bit in the golden era of gene regulatory networks. And the reason is that we can have cell type specific expression profiles and also cell type specific chromatin accessibility profiles, meaning that this combined with a good catalog of transcription factor motifs, such as the one of CAX2 that Victor has showed before, we can try to infer with much better accuracy regulatory relationships. Okay? So that's what Cell Oracle does. This is the strategy of Samantha Morris, and this is a strategy that we have adopted to make gene regulatory networks in our system. So we are going to build gene regulatory networks specific of each type of radial glia cells in the in vivo human brain development, and also in the different phases of neurogenesis, and also in gliogenesis. Okay? So to do that, we need data sets that combine RNA-seq and ATAC-seq as a proxy for putative regulatory elements, right? Chromatin accessibility regions in a cell type specific manner, single cell. So we got this mid-fetal primary data from the human cortex, from Turbino, from Pascal lab, and then we painfully, reprocess all this data to get the cell populations that we need, because every lab that has its own effort, but sometimes it's not exactly what you need. So here is the final annotation that we are using, that we did for months. So essentially we have radial glia cells, truncated radial glia cells, as Victor has explained before, astrocytes, OPCs, glial progenitor cells, intermediate glial progenitor cells, intermediate neuronal progenitor cells, and so on and so forth. So we have this axis of radial glia maturation then, and then we also have this axis of neurogenesis that we can interrogate. So we constructed these networks in a cell type specific manner. So you have in the y-axis all the cell types for which we have a network. And it is a global network conveying all the transcription factors that are expressed in that cell type. And then, since we have that, we can cross it with our transcription factors associated with disease, right? And then make questions in terms of topological measures of the network. For example, is my transcription factor associated to a particular disease highly connected in the network? It is a hub, it is a peripheral node, and we can make all this kind of topological measurements of the network. If we do that, we can have a glimpse of the role or the relevance of this transcription factor in each cell type in terms of connectivity, right? So by doing that, we see, for example, that MEF2C it's really highly central or really highly connected in glutamatergic neurons, but also in some autoradial glia cells in the early phases of autoradial glia cells maturation, or KLF6 is really central essentially in many, many cell types across these different axes. Okay, so once we have these regulants, we can make the opposite question. We can say, is there any of these regulants that is agnostic of disease? It's just the gene regulatory network of the entire cell. Is any of these subregulants orchestrated by each one of the transcription factors expressed in this cell type enriched in gene sets, in disease gene sets? And then the answer is yes. So we can make for each one of the disease what are the top transcription factor whose targets belongs to a given disease gene set, right? And then we have, for example, for ASD, in ventricular radial glia cell are enriched in the regulon orchestrated by PO3F4 or ARX or BCL3. In the case of schizophrenia, ventricular radial glia cells are, genes from schizophrenia are enriched in regulons in ventricular radial glia cells orchestrated by MEF2C or NPAS2 and so on and so forth. So it triggers, it gives, hypotheses on the transcription factor that can be upstream of these pathological processes. Okay, now that we have a network, I want to explain what this network means in terms of these edges. So essentially, the network models how much the difference of the expression of a transcription factor influences the expression of the target gene. Of course, one gene can be regulated because we have the chromatin accessibility landscape. It can be bound by multiple transcription factors. Then, essentially, every gene is the victim of a multiple linear model in which every edge connecting a transcription factor with that gene is a linear model with a coefficient, right? So since we have these coefficients, essentially it means that we can take out one of these genes or overexpress one of these genes and recalculate the network and what's going to be the transcriptome after one iteration or after two iterations and so on and so forth. And this is what Cellular is doing for us. And then once we have the new transcriptome, the one that is the result of these iterations, we can compare this new transcriptome with the cells that we have in the system and say, okay, I was originally a ventricular radial glia cell early, but look now, I look now like an intermediate progenitor cell, or I look like a neuron, okay? So we did this perturbation for each one of the transcription factors. And here we have our annotation of cell types for the radial glia progression coupled to gliogenesis. So we have ventricular radial glia early, late, ending up in truncated radial glia cells. But then we also have these autoradial glia cells. And then we have intermediate glial progenitors going either to OPCs, to oligodendrocytes, or to astrocytes. So now, in here, in each one of these cells, we can project what is the direction of change after perturbing the expression of KLF6, after essentially removing KLF6 from the network. Okay, it's like a in silico knockout. And you can see in this other graph plot here how the different populations have changed after the perturbation. And we can see, for example, that after perturbing KLF6, glial progenitor cells or autoradial glia cells become intermediate glial progenitor cells, and glial progenitor cells become oligodendrocytes. So we have this prediction of what's going to be the effect based on the inference of the gene regulatory network. So we did that systematically for all the transcription factors in the network. And then by doing that, we kind of summarize how much each cell type is perturbed and to what other cell type it transitions after knocking out the transcription factor. And then we kind of size the role. We can observe that some transcription factors, even if they are highly expressed in excitatory neurons, they can have multiple critical phases in which they are, exerting an effect, perturbation on the transcriptome at different time points and different phases of radial glia maturation or neurogenesis. And we also can size this effect. And we can see also that the expression doesn't really have a sync with the role in the network, and one transcription factor can be very hub in one cell type and very peripheral in another cell type. So the conclusions of my talk would be that differentiation trajectories coupled to this multi-ohm data will allow for the first time for meaningful gene regulatory networks inferences, that once you have a good inference and a good gene regulatory network, you can have powerful in silico predictions. And by doing so, we identify new implications for genes that were previously associated to other more mature terminally differentiated excitatory neurons cell types, and identify multiple critical phases in specific cell types, which can point to vulnerability phases, even in brain organizing centers. And this, on top of that, is essentially a general resource for making testable in the lab hypotheses. So with that, I want to finish to say that this project has been done in collaboration with the lab of Pasco Rakic, with Nicola Micali, and this is essentially the work that has been done by Joel Mato Blanco, a PhD student, brilliant PhD student in my lab. So thank you very much. I'll take questions.
Any questions for Gabriel?
Gabriel, I think that was great. And you left me in your last slide with the question of which is the champion of transcription factors making the most influence.
Well, it depends on the specific comparison that we want to know. So we have ordered, for example, which is the one that depletes the most autoradial glia cells. Well, you have a list. Which is the one that depletes the most intermediate progenitor cells, which is it's TBR2, by the way. So, and, you know, so we have a list for each one of the transitions that you can, this is a really complex, and we have in terms of the number of comparisons that you can do. So we have also I just show you the radial glia maturation, but also the neurogenic axis. And then you have different types of neurons. If they go forward or they go in the reverse.
Sure, but if you give each of these changes a score, you can have a global score of...
The score is based on the score is based on the how many cells, like, do you eliminate after the perturbation. So if you had from 100% radial glia cell, you have whatever number of radial glia cells. If all of them goes away after the perturbation, this would be the top highest score. And then the top highest score is for genes that are really like PAC6 for the glia cells or TBR2 for, you know, genes that we know that are really identity, but actually they happen to be predicted from the network, right? So it's interesting, I mean, because this is just how the transcriptome is perturbed after the expression. So it should be agnostic of markers, right?
Very nice talk. I have a super naive question, and it's regarding because every time there are more and more data that is published from single cells. And I was wondering whether your system is being updated or whether, depending on the raw data, the network could be also changed in silico. So what's your...
So you mean if we can repeat the same approach every time that a bigger and better data set it appears? I mean, absolutely. I mean, at some point, this is a matter of every day, you know, you have millions and millions. It's a computational problem of being able to integrate all these data sets, and of course, the better resolution, the best. I mean, yes, and then because it's important that, because when we have a small number of cells for some of these populations, the inference are worse. So we need balanced data sets, and for that we need millions of cells. This has a lot of caveats because we are using, you know, this data set. In the future, it's going to be better.
Actually, I have a question. So I know that you focus on neuroprogenitor cells, but in one of your slides where you analyze across different cell types, there were some mesodermic lineages that just pop up in diseases. So have you checked changes in signatures in microglial cells? I mean, that must be also important during cortical development. I mean, they can remove some of intermediate progenitors, then on synapse refinement.
Yes.
So have you ever checked those?
No, we didn't focus at all in microglia in this project, but I totally agree that, this...
like most of these diseases also produce inflammation, right?
Absolutely. We should check if what proportion of these gene sets are also expressed in microglia. Yes, why not? Yes, it's a good idea.
Any more questions? Okay, so let's thank again Gabriel, Victor, and Dani. Thank you very much.